Welcome back to another explainer. How do we actually tell if a groundbreaking medical therapy works or if the data is, well, just kind of lying to us? Today, we're investigating the world of regenerative medicine, specifically looking at renal cell therapy meta-analyses. Our goal here is to extract the true reproducible biological signals from what is, frankly, incredibly noisy data. We're going to diagnose the data flaws, make sense of the statistical variances, and define some rigorous solutions to really elevate future clinical research.
So here is our roadmap for today. 1. The promise of cell therapy. 2. Acute versus chronic models. 3. The heterogeneity complication. 4. Sources of experimental variance. 5. Filtering for reliable signals. And finally, 6. Best practices for the future.
Okay, let's dive right into this. We are taking a really complex biological mechanism and trying to make sense of scattered, chaotic data points based on a huge evaluation of cell-based therapies in renal injury. We're specifically focusing on meta-analyses that evaluate acute ischemia reperfusion models versus chronic kidney disease models using murine or mouse data.
This basically sets up the three pillars of our entire discussion. First, we've got a consistent biological response, like we know for sure these therapies are doing something positive. But we face this massive heterogeneity divide, where acute models just throw off so much statistical noise compared to the much more stable chronic models. And finally, the path forward absolutely requires strict data filtering to isolate the true clinical signals. Let's unpack that first pillar.
Section 1. The promise of cell therapy.
Before we get into all the heavy statistical critiques, we really have to establish the baseline truth of this research. Mesenchymal stem cell, or MSC, therapies universally show signs of working in renal models. Across wildly different parameters and experimental groups, researchers consistently track a real biological improvement. We're seeing a highly reliable 5 to 8 point reduction in critical renal damage biomarkers compared to control groups. Whether you look at blood urea nitrogen, creatinine, or markers of cellular distress like apoptosis, fibrosis, and oxidative stress, the signal leans distinctly toward improvement.
In fact, researchers discussing this data in the lab summarize it perfectly. They noted, they all show reductions. It's a consistent overview of lowering BUN and creatinine across the board. And this perfectly reflects the initial optimism in the field. When you see numbers dropping across all these vital markers, it's thrilling. But, and this is a really big but, as we're about to see, that optimism hits a brick wall when we actually look at the underlying statistics.
Which brings us to Section 2, Acute versus Chronic Models.
So, if the therapies are universally showing reductions, why does the scientific community have such different levels of confidence in the data? Well, it all comes down to the structural differences in the models used to evaluate those biomarkers. When we break down the scope of these meta-analyses, we're basically looking at two completely different worlds. On the acute ischemia reperfusion side, the rapid onset and immediate treatment windows create a massive volume of available literature, but incredibly high noise. On the chronic kidney disease side, there are fewer published papers because, honestly, long-term evaluation is demanding, but the data sets are highly stable and consistent. When you compare the data profiles head-to-head, the difference in statistical variance, or what we call average heterogeneity, is staggering. In the chronic model, the variance is consistently under 25%, which gives us a really high level of reliability and confidence. But the acute model? The average heterogeneity is often hitting 60%, 88%, even up to 92%. That means the data from these acute studies are wildly inconsistent with one another, and our confidence level plummets.
The crucial takeaway here is super simple. High literature volume does not equal high data quality. A massive pool of acute data where the variance exceeds 88% just creates this wall of statistical noise that drowns out the clinical signal. On the flip side, fewer chronic papers with under 25% variance yield a much clearer, truer picture of what's actually happening.
And that leads us to Section 3, the heterogeneity complication.
This brings us to the climax of the data problem we're unpacking today. What happens when mathematical heterogeneity actually obscures the biological truth? There's a massive divide in mathematical standards here. On one side, we have what's referenced as the Loux standard, aiming for a heterogeneity or I-squared value of under 25% for high reliability in a reproducible signal. On the other side is the acute reality, massive variance between 60 to 92%, because unlike human trials, animal studies lack standardized baselines. Averaging data with 90% heterogeneity is incredibly dangerous, mostly because it creates mathematical noise that can completely misrepresent the actual biological effect.
So you gotta ask the critical question, and it's the exact same skepticism raised by researchers analyzing this data. Why is the acute dataset so incredibly inconsistent? What is driving this 90% variance? Let's round up the suspects.
Section 4. Sources of experimental variance.
It's time to investigate the specific variables causing this massive chaos in those high-volume acute models. When you map out the acute parameters, you immediately see a glaring lack of standard protocol. Ischemia duration fluctuates anywhere between 30 and 60 minutes. Treatment timing is completely scattered. You've got pre-ischemia, during reperfusion, well after. And the dosage spectrums are incredibly wide. When your experimental foundations are this different, you inject massive structural variance into your meta-analysis before you even look at the results.
And this brilliantly illustrates the sheer chaos. There are 4 core discrepancies plaguing this data pool. First, the animal strains vary. Researchers are using different models, like SD versus BIS strains. Second, the cell sources differ wildly, mixing human-derived MSCs with animal-derived cells. Third, the evaluation technologies used to measure outcomes aren't standard at all, mixing NTA and TM techniques. And finally, you have the dosage disconnect.
Let's really think about that last point for a second. Mixing a 20 microgram outcome with a massive 250 microgram outcome into the exact same mathematical average. That's literally like throwing an apple, an orange, and a shoe into a blender and trying to calculate the average flavor, right? It completely dilutes the clinical findings. The logic of combining these highly disparate inputs into a single statistical average is just deeply flawed.
Section 5. Filtering for reliable signals.
Okay, so we've identified the noise and diagnosed where it comes from. Now let's talk solutions and demonstrate how researchers must ruthlessly clean this data to find actual scientific value. Here's the proposed method, the reliability filter. You take the raw literature data and you apply a strict under 25% heterogeneity filter to the acute data pool. You must explicitly discard or heavily downweight the outliers. Why? Because including highly disparate data just because it exists artificially inflates the baseline efficacy. By recalculating the averages using only the highly reliable studies, the true biological signal is finally revealed.
But wait, there is a massive mechanistic caveat we have to acknowledge here. Recent literature confirms that MSCs are not static, dumb drugs. They are dynamic signal transmitters. A healthy parent source transmits regenerative, anti-inflammatory signals to the target tissue. But if those parent cells are compromised, damaged, or stressed, they actively transmit pathogenic or apoptotic signals. This means they can actually exacerbate the renal injury rather than fix it.
As noted in the lab transcripts analyzing this exact issue, MSCs are not always good. If you inject compromised cells, they will transmit bad signals and the outcome will definitely be bad. This really grounds our statistical data in biological reality. Efficacy isn't just about dosage amounts or ischemia timing. It is fundamentally tied to the quality of the source material being injected.
Section 6. Best Practices for the Future. We've journeyed from recognizing universal efficacy to uncovering some pretty deep statistical flaws. So let's translate these rigorous critiques into actionable takeaways for the field. To ensure future meta-analyses actually guide clinical success, researchers must adopt these 3 mandates. Number 1, standardize variables. Future animal studies must align on dosage, like sticking to 100 to 250 microgram range, and standardized ischemia duration to allow for true one-to-one comparisons. Number 2, filter ruthlessly. Do not average noise. Meta-analysis must explicitly discard studies with over 60% heterogeneity. And number 3, prioritize source quality. Acknowledge the signaling nature of cell therapies and standardize the validation of the parent cells before you ever evaluate the final outcome.
Which leaves us with one final pretty massive question. If we stop accepting sheer volume as a substitute for rigorous quality and actually apply these ruthless filters, how many failed therapies might actually be hidden successes? True scientific value comes from data reliability, not literature volume. Keep questioning the noise to find the signal. Thanks for joining me on this explainer, and I'll see you next time.